Our second House Scholar is Dr. Arianna Krinos, who earned her PhD in 2024 from the MIT Woods Hole Oceanographic Institution Joint Program in Oceanography and Applied Ocean Science and Engineering. After graduating, Arianna joined Brown University's Center for Chemical Currencies of a Microbial Planet, where she is a postdoctoral researcher. Arianna's research focuses on the dynamic ecological roles played by diverse microbial communities in marine environments, which play critical roles in global environmental cycles. Arianna has enabled substantive breakthroughs addressing many of long-standing challenges for microbial ecology. One example of this, environmental samples usually include novel species, strains, etc., so the community has long struggled to identify what species, genuses, and so forth might be present or absent in a sample. Annotating by aligning sequences to existing reference databases creates potential for huge biases. Arianna proposed a major shift in perspective, moving the community away from sequence alignment and towards a clustering approach, which both provides more insights and is also more robust to these potential biases. A portion of this work was conducted during Arianna's 2 practicums at the Joint Genome Institute at Berkeley Lab, where she leveraged JGI's unique scale of biosequence data and its HPC capabilities. Arianna's contributions to her institutions and communities are similarly impressive and indicative of her character. She mentored 4 undergraduate researchers during her PhD. She has both sought out and created opportunities to increase her research community's knowledge and skill with computational methods. These include leading software carpentry workshops, lecturing or co-teaching multiple courses and seminars in the MIT Woods Hole Joint Program, and teaching middle schoolers and high schoolers, going so far as to design and teach a high school course on Bayesian pattern analysis. Dr. Arianna Krinos is an extraordinary computer scientist and a polymath, able to drive research at the intersection of computing and the life sciences. She is also a gifted and dedicated teacher, as well as a scientific leader. She exemplifies the technical excellence, scientific leadership, and outstanding character that marked Fred Howe's career, and which this committee recognizes with this award. Let's congratulate Arianna.
I'm very excited to speak with you all today about my research, understanding phytoplankton and marine ecosystems and using various computational tools. I'll take you on a bit of a tour today through some of the various computational methods that I use and try to contextualize them with the reasons why we care about marine ecosystems and specifically about phytoplankton and other microbial eukaryotes within those ecosystems. So to begin by motivating this, the ocean is a vast and important resource, a very important part of Earth's biogeochemical cycles. The ocean is important because it sequesters carbon dioxide into carbon biomass, which is really important in global climate and the cycling of carbon. It's also important economically because it's a source of economic value through fishing and food security worldwide. And it's also biomedically important. The ocean is frequently a source of new antibiotics, new biomedical treatments, and other breakthroughs in medical science. The ocean covers about 70% of Earth's surface, so for that reason alone, it's critical to understand how the ocean works and how it determines aspects of our lives, like weather and climate. And within the oceans, microbes are a critical component. In every drop of ocean water, there's on the order of 10 to the 5th microbes in each drop. And summing that biomass together, the total microbial biomass in the Earth's oceans is on the order of 10 to the 17th grams, which is the mass of around 43 billion elephants, or put another way, it's more than 550 times the biomass of all humans that presently live on Earth. So microbes in the ocean are a significant biomass contributor, and they're playing diverse ecosystem roles. Within microbes in the ocean, microbial eukaryotes are also important. We frequently think of microbes as bacteria. Microbial eukaryotes are a big part of the biomass of all living things on Earth. This quadrilateral here is showing microbial eukaryotes in the context of all living biomass across ecosystems on the planet. And microbial eukaryotes are important because they play diverse ecosystem roles. namely what or whether they're eating in an ecosystem, how they're playing a role in the food web, determines how they're contributing to nutrient cycling and how they're hence contributing to those global biogeochemical cycles that we care about from a climate perspective. So phytoplankton, we like to refer to them as forming the base of the marine food web because they take carbon dioxide from the atmosphere and assimilate that into carbon biomass that then could be exported. It could sink to deep depths. And heterotrophic protists consume other organisms, so they might link the numerically abundant bacteria to higher trophic levels or perform carbon transfer in order to take that carbon that's being produced at very small sizes within that microbial world and make it available to larger organisms. So to make that more concrete, I'll talk about some microbial eukaryotes during the talk today, so I want to just mention what they look like, who they are. Cryptophytes are an example of a prey species with mixed nutritional modes, so they can both photosynthesize and consume other organisms, which is one of the things that makes these organisms unique, but also difficult to track, which is one of the reasons why we need computational modeling approaches. Diatoms are photosynthesizers. These are the classical sort of late winter, early spring bloom formers that can assimilate a lot of carbon and exploit really high nutrient conditions right after winter sort of ends and temperatures get high enough for these organisms to start growing quickly. Ciliates are an example of heterotrophic protists that consume other organisms, form links in the food web. Coccolithophores are calcifying photosynthesizers that form these calcium carbonate sheets and again are really important to carbon cycling. We'll talk about them more later. Radiolarians are predatory protists, so they're heterotrophs, but these are also an example of some of the unique biology that can be difficult to simulate. In that these are heterotrophic protists, they're pretty large, but they have photosynthesizers living on the surface of the cell. So they're symbiotic, and that plays a role in how we're able to actually track where the carbon's going and how it's being used. So in order to study microbial eukaryotes in the ocean, we need tools that cross scales. We do need laboratory experiments, either ones we conduct ourselves or using repositories of data online. We need to understand how microbial eukaryotes are responding to controlled laboratory conditions. So if we hold something constant, how are they responding? We also need field observations to measure how microbial eukaryotes are behaving in their natural environment, in this case in the ocean. And we need modeling approaches, mathematical representations of how microbial eukaryotes are functioning. This is really important because of those scale questions, those scale features that I've highlighted a few moments ago, in that we can never really measure comprehensively where the microbial eukaryotes are, how abundant they are, and what they're exactly doing. We need to be able to scale that up using these simulation approaches. Increasingly, genetic sequencing approaches are enabling us to link laboratory physiology experiments to field observations, because we can sequence something in the laboratory and look for that exact same signature in the field. So we need to be able to both track what's happening in a controlled laboratory setting as well as what's happening in situ. We can try to get an approximation of that using genetic sequencing, which I'll talk about in more detail in a moment. We need both these laboratory physiology experiments as well as field observations in order to do any modeling. We need laboratory physiology experiments to parameterize our models, and we need field observations to validate them. So for genetic sequencing approaches, the types that I use are nucleic acid sequencing approaches. So I like to put the general central dogma of biology up here just to explain what that means. We have DNA stored in the nucleus. That's what we refer to as genomics. So we have DNA that is all the instructions that a cell knows how to make any protein that's in its repertoire of possible proteins that it can produce. We have a subset of those sequences that might be converted, transcribed into RNA. So these are active signals that are being used to respond to current environmental conditions that then get translated into proteins that do the work within the cell. And within microbial oceanography, we might be talking about either single target omics, so we might be talking about a laboratory experiment where we sequenced one thing, one species at a time, or meta-omics. So this is a really powerful tool where we're just going to sequence everything that's present in the ocean water and then use computational approaches to back out who was there and what they were doing at a given time. And database approaches are really essential, critical for linking single target omics to multi-omics. That was something that was mentioned in the introduction. In that the inadequacy of databases to represent everything that's present in the microbial communities can really hamper our ability to understand ecology and biogeochemistry. And we have a lack of tools that are designed specifically for these kinds of problems, for environmental microbial eukaryotes in places like the ocean. So one aspect of my work has been developing different pipelines, computational tools for processing, sequencing data from environmental communities. This is a tool for assembling and annotating metatranscriptomes, so community level expression data. Transcriptomics is sequencing the RNA, forgot to mention that on the previous slide. And then this is a pip and conda installable package for annotating taxonomy and including using some of those clustering approaches to identify totally unknown, previously unseen microbial eukaryotes. So this, again, is available online, and it's been a resource for the community that are trying to study these microbial eukaryotes, both to download database resources as well as to expand the potential of what we're able to annotate. So to switch gears a little bit, I'll talk about one specific microbial eukaryote, Gephyra huxleyi. It used to be referred to as Meliania huxleyi if you've been in an ocean science class. This is a calcifying species of phytoplankton that forms globally significant blooms. So this satellite image here is showing off the southwest coast of Ireland a bloom of Gephyra huxleyi, a coccolithophore, where you can see that milky white color in the ocean is a bloom of coccolithophore. This is satellite detectable, so we can see it from space. We could use that to track something about where the coccolithophores are concentrated, though we can't necessarily tell who exactly they are from that information. This is an electron micrograph of the surface of a coccolithophore, so those are those calcium carbonate sheets. They're important because they have a complex relationship with the carbon cycle, so they're really important in understanding global ocean carbon cycling. Because the process of photosynthesis takes up carbon dioxide, turns it into carbon biomass, but the process of calcification releases carbon dioxide. So it's now a complicated equation for coccolithophores with respect to whether they are consuming more carbon dioxide, taking more carbon dioxide up, or emitting more, depending on how heavily they're calcifying. And also, many different strains of this species are maintained in culture. So genetic diversity is something that I think a lot about in terms of present diversity and also in terms of possible future diversity. So evolution and response to future environmental change. And interspecific diversity is important because a combination of these factors, genetic factors as well as environmental factors, can lead individuals within a species to dramatically vary. We know this from humans. This is human height distributions. All humans have a slightly different height, and that is controlled by a combination of genetic and environmental factors. We also know this from dogs. All dogs are members of one species, but different breeds of dogs are different because of intraspecific variation. And phytoplankton are not immune to this idea of intraspecific diversity. So intraspecific diversity can lead to distinctive ecosystem roles, and this can be what enables a coccolithophore like Gephyra huxleyi to live all across the ocean, not just in one specific type of environment, but different specific types, different ecotypes of this strain might have different copies of key genes that enable it to live in very different environments. So one way that I approached or I tested this idea of intraspecific diversity, and specifically in the context of thermal response, was using a series of experiments where we exposed multiple strains, in this case 12 strains of Gephyra huxleyi, to different environmental temperatures according to their reasonable expected range in situ. So this setup, this thermal block, has a circulating water bath that keeps temperatures cooled on the left and a heating element that warms up temperatures on the right, which sets up a gradient of a difference of about 1 degree Celsius between adjacent columns of this thermal block. And so we're able to test many temperatures at once and to expose these strains of Gephyra huxleyi, originally isolated from all over the global ocean, from close to Antarctica to close to the equator, to different temperatures over a very long period of time. So this was about 2 months of thermal acclimation that these strains were exposed to. And this is important because thermal physiology determines the distribution of phytoplankton. Phytoplankton growth rates are highly sensitive to temperature, and tipping points in temperature or changes in temperature regimes or the specific tolerance of phytoplankton to those temperatures can determine the range and distribution of phytoplankton, determine the formation and termination of phytoplankton blooms, those large accumulations of phytoplankton biomass that can affect carbon export, their ability to resist viruses, and their macromolecular composition, so their relative composition of major macronutrients like nitrogen and phosphorus, that again will determine both their palatability to higher trophic levels, so that sort of food web transfer, as well as export of different nutrients to the deep ocean to be stored over long periods of time. So our question is whether strain identity might determine thermal response. In other words, if we have multiple strains of Gephyra huxleyi, whether we might have some strains that are more like a husky dog, really tolerant to cold temperatures, and others that are more like an Australian cattle dog, more persistent in warmer temperatures. So in order to do this, I use the Darwin model. This is an ecosystem layer on top of a global biogeochemical model. So an Earth system model that simulates the physics and the chemistry of the global ocean. And then in each grid cell, there is a biological module, an ecological module, where we both have very different phytoplankton functional types, as well as, for example, predators that are consuming those phytoplankton. And we're able to encode information about the traits, so the specific characteristics that differentiate one phytoplankton type from another. For example, we might differentiate diatoms from another group of phytoplankton via their ability or their need for silica, some mineral that they require. So from those experiments, what I get are these thermal performance curves, which are the currency that I can use to tweak those traits within that Earth system model, so within that global biogeochemical model, MIT GCM. On the x-axis, this is temperature from the experiments, and the y-axis is estimated growth rate in per day. An estimated growth rate about 1 per day means that the population is doubling itself once every day. And from these curves, I can extract the thermal optimum. This is the temperature at which growth rate is maximized. And also the thermal niche width, so the range of temperatures at which the growth rate is non-zero. So what temperatures are going to be survivable for each one of the strains. And from these experiments, I noticed that the thermal niche width varied a lot between strains. Here's 2 different strains that have very similar thermal optimum value, very similar temperature at which growth rate is maximized, but a different range of habitats over which they can survive. And this is not something that is encoded in this Darwin ecosystem module with MIT GCM. We don't, in other words, have the ecological concept of specialists, organisms that are able to tolerate a relatively narrow range of temperatures, but potentially with some kind of a growth rate advantage, and generalists, organisms that are able to tolerate a wide range of temperatures, but potentially with some kind of growth rate penalty. So in order to address this, I modified the base simulation of the Darwin model and added a variety of thermal niche widths to that simulation. So now, instead of having a single width of the thermal reaction curve at each temperature of the thermal optimum value, I now have many different thermal niche widths that are simulated and a modest penalty for adopting a wider range of survivable growth rates. So from the Darwin model output, we're able to assess the possible drivers for differences in the ecology, differences in the survivorship of these different ecotypes that we've now encoded into the model, this additional complexity that we've introduced. One way to look at that is to first look at the modeled variability in temperature, so standard deviation of temperature here assessed across the global ocean in model space. And if we zoom in, for example, on the east coast of the United States, we can see that the standard deviation of temperature is higher closer to the coast and starts to decrease as you get further from the coast. So we have higher thermal variability closer to the coastal habitats here, which makes sense. And when we look at the modeled thermal niche width, so the prevalence of generalists versus specialists in model space, we also see that thermal niche width is higher in those more thermally variable environments closer to the coast. However, if we look across the whole planet in terms of the average thermal niche width, so higher thermal niche width being more generalists and lower being more specialists, we have some imbalance. So higher thermal niche width is present in the northern hemisphere. And we also have other oceanographic features that are driving the prevalence of specialists versus generalists in this model ecosystem. In other words, generalists and specialists do tend to correlate with regions of higher thermal variability in ocean dynamics, versus being broadly predictable across latitudes. And this is important because the prevailing hypothesis in ecology is that thermal niche width is going to increase with increasing latitude, because in terrestrial environments we do tend to see that thermal variability increases as latitude increases. So in places that have more seasonality, more seasonal variability, there's going to be higher thermal variability and hence more of an advantage of being a generalist. So in order to take this a step further, I set up a bivariate normal distribution, which I constructed using the minimum and the maximum values for each of the ecotypes that were present, that were able to survive in each modeled grid cell within the Darwin model output, in order to calculate a probability of the survival of each of the strains that I had worked with in the laboratory. So using this bivariate normal distribution, I was able to calculate a probability of survival of each of the strains from the experiments. So for strain 6071, this diamond is showing where it was originally isolated from, close to Antarctica. It has highest probability of survival at the poles, which means that this strain of Coccolithophore is more like our husky dog, more tolerant of cold temperatures. And we can see for a strain like 3492, which was isolated here close to the coast of Japan, it's most probably found based on this Darwin model habitat, this Darwin model framework for habitat prediction, closer to the equator. So this, in other words, is our Australian cattle dog among coccolithophores. We can do this same thing for all of the strains that we tested in our experiment, so we can evaluate how likely it is that each of these strains will persist in the current ocean as well as project in the future, how likely it is that this diversity will be maintained under future environmental conditions. So in summary of this part, laboratory experiments show us how trait variation within the phytoplankton can determine their distribution, their biogeography in the global ocean. The trait-based modeling framework of the Darwin model enables us to encode traits into a computational modeling framework to couple biology with physics and chemistry, those larger scale dynamics of ocean ecosystems, to put that information to work where it counts in understanding biogeochemistry, nutrient export, other factors that we're interested in terms of the global relevance of phytoplankton and other protists. We also know that higher temperature is linked to higher stratification, so higher nutrient limitation. And so the next thing that I'll show is how we can use genetic data to dig deeper into this idea of interspecific variation, but also in general mechanisms for dealing with both temperature and nutrient limitation in ocean ecosystems. So in order to illustrate this, I will show a data set that I collected during my PhD. So this was in collaboration with the Center for Coastal Studies and the Massachusetts Water Resources Authority. This sampling has taken place overall since 1992 in Cape Cod Bay, originally chartered because of the movement of a wastewater effluent site from Boston Harbor. Monthly, there were CTD casts, so this is conductivity, temperature, and depth, analysis of inorganic nutrients, as well as visual identification of phytoplankton and zooplankton through microscopic counts. And from 2021 to 2023, we augmented this data set. So I went out monthly and collected molecular data, so 18S and 16S rRNA gene sequences for identifying, barcoding bacteria and phytoplankton, as well as metatranscriptomic sequencing, so community-level expression data for identifying the potential physiological mechanisms as encoded in the production of RNA for phytoplankton blooms that are occurring over seasonal cycles in this location. And having access to this really long-term data allows us to probe longer-term trends that might be occurring as a result of local environmental change. In Cape Cod Bay, this is a decadal shift of a decline in nutrients that we can see from the fact that we have this very long-term data set. And so this, after 2010, there was a reduction in average nutrient concentrations, likely driven by ocean circulation in the region. And what we can also see from these long-term data of the microscopic phytoplankton counts is that around 2010, I noticed that there was this increase in coccolithophores that were recorded in this location. So whereas before 2010, we really never found coccolithophores in this location, after 2010, we start to. This is really interesting because coccolithophores are not frequently found close to the coast. They're more likely to be open ocean and to live in really low nutrient environments. And this corresponded with the decline in phosphate concentrations after 2010, which is significant because coccolithophores are expected classically to thrive under low phosphate concentrations, and past models have predicted that coccolithophores will be most successful under phosphate limitation, which leads us to our question of whether phosphate stress can relate to coccolithophore success in coastal ecosystems where they previously were not abundant. And so to motivate this, from a biochemical perspective or from a biological perspective, there are three main ways that organisms deal with phosphate stress. The first is potentially to take up more inorganic phosphate than everyone else. This is the uptake mechanism. We can take organic phosphates and cleave them from organic matter, so we can use that mechanism to deal with our increased need for phosphate. And we can also remodel our metabolism to get rid of the need of phosphate in the first place. So an example of this is that we can take phospholipids, which are commonly used in the membranes of cells, and replace them with sulfolipids and galactolipids. So within Jafira capsulohuxley, I could look within the metatranscriptomes at the pathway responsible for this substitution of phospholipids with sulfolipids and galactolipids and see that there was increased expression of the genes that were responsible for both converting phospholipids to sulfolipids and phospholipids to galactolipids. So what this tells me is that what the Jafira capsulohuxley, the coccolithophores in this environment are doing is getting rid of their need for phosphate in the first place, so remodeling their metabolism. And comparing coccolithophores within this data set in these metatranscriptomes, where again we have information about the entire community of microbial eukaryotes all at once, we can look at coccolithophores and compare them where they're abundant at low phosphate concentrations to organisms that are abundant instead at high phosphate concentrations and see whether this is unique, whether this metabolic mechanism that they're using is specific to coccolithophores or whether it's something that everybody uses no matter what when they're subjected to phosphate stress. So we compare the coccolithophores in this case to cryptophytes and diatoms. These are organisms that are abundant in spring, that is, that have a different way of dealing with phosphate stress in general. And we can see here that these genes that are responsible for dealing with phosphate limitation are pretty universally upregulated, so they're increased in expression, they're used a lot more heavily under low phosphate concentrations in Jafira Huxleyi, but generally they are not used more under low phosphate concentrations by cryptophytes and diatoms, indicating that this is a unique mechanism that coccolithophores are able to use that cryptophytes and diatoms are not. So this impacts the way that we're thinking about models because something like the trait-based modeling framework of the Darwin model enables us to accommodate this variability between different biological species, different functional types that we might want to simulate in that global ocean context. And we can also relate this back to those experiments. So in addition to measuring the thermal performance curves of the strains in those experiments, I also sequenced transcriptomes, so got gene expression data for each of those different strains, different types of this one species of coccolithophore. And in that transcriptome data, so one species instead of many species at once, I can look for the same genes, so those genes that are responsible for substituting galactolipids or phospholipids with galactolipids, that is, in this case in the data set. And I can see that there's diversity within these strains. So not all strains are doing the same thing with respect to this substitution mechanism under temperature limitation. So again, here we are not measuring nutrient limitation, but we know that there's some interplay between these two things. So temperature limitation and nutrient limitation are linked. For our warm adapted strains, so our strains that we found in warmer environments, we have more expression, so upregulation of this gene for galactolipid substitution at higher temperatures. Whereas for our cold adapted strains, we have more expression, so more use of this gene responsible for substituting phospholipids with galactolipids at lower temperatures. So we have an opposite response based on the adaptation state of the strain, which is really important for thinking about ecology and global distributions. And for this strain that's a little bit in the middle, we have some copies of this gene that are more regulated and more highly expressed at higher temperatures and some at lower temperatures. So there's interspecific diversity in how this gene is being used under temperature stress. So in summary, phytoplankton ecology can be incorporated into global models using trait-based approaches that enable us to simulate what makes phytoplankton different, which is really important for simulating how they play roles within the ecosystem as well as in global biogeochemistry. Our experimental evidence suggests that there's tremendous diversity that already exists within the phytoplankton, which leads to lots of differences in traits that should be encoded into models and might in the future encode evolutionary potential that is of interest to us as we're studying global environmental change. These traits are further understood, disentangled via molecular data, which are aided by these computational approaches and pipelines that enable us to use these vast amounts of data from across the global ocean, which are increasingly being generated and uploaded into online data repositories. And integrative computational approaches are critical for predicting global change in addition to assessing what genetic diversity already exists and how it can be used by these organisms in responding to a changing environmental context, like we saw in Cape Cod Bay. I'd like to thank many people who were involved in this work, and also, of course, to thank the CSGF program and the selection committee for inviting me to speak to you all here today. And with that, I am happy to take any questions with the time that I have.